Take a deep breath in. Now breathe out. It's refreshing to get oxygen in your system, right? But who made that oxygen for us? Well, plants, of course. Through a process called photosynthesis, the leaves of plants take carbon dioxide, water, and sunlight, and use them to make food for themselves, in the form of sugar, and make oxygen to release into the air. Photosynthesis has been happening in nature for billions of years. Today, scientists study this process and take inspiration from how plants do it, and they are trying to find new and better ways to gather the energy from the sun and store it in fuels or electricity which we can use. You've probably heard before of things like solar cells or solar fuels. These are examples of what we call artificial photosynthesis. Today, we are starting from the first step of artificial photosynthesis research, studying natural photosynthesis as it happens in plant leaves. Because it is hard to study something that you can't see, we are going to find a simple way to observe it in action and measure how fast it goes. In order to do photosynthesis, plants only need 3 ingredients: water, light, and carbon dioxide. We can easily give the leaf these 3 ingredients by using a simple lamp and a solution containing sodium bicarbonate, which is carbon dioxide dissolved in water. If we take a baby spinach leaf and we place it on water, it floats. This is because one of its tissues, called mesophyll, contains a spongy structure that traps air and makes the leaf less dense than water. If we replace that air with water, we can first make the leaf sink, and then use the spongy tissue to trap the oxygen released by photosynthesis until the leaf starts to float again. To make things simple and fair, we are not going to use whole leaves. Instead, we are going to cut out small discs of the same size. When we do this, we must be gentle and try to use the green bits of leaves without veins. Let's cut out 10 discs. We can now put the discs in a syringe together with some of the solution we prepared before. Gently pulling the plunger while holding the thumb against the hole will lower the pressure and pull the air out of the leaf discs, allowing our solution to fill in the gaps and eventually causing the discs to sink. Once the discs are in our beaker, we can add our last ingredient, light. We now just need to start a timer, wait for the discs to start floating, and write down what we observe. After every minute, we should write down how many discs are floating, then gently mix the solution to stop the discs from getting stuck on the walls. What's happening inside the leaf discs? If you've already visited Holly's experiment, you should know everything about electrons and their interaction with light. What happens in a leaf follows exactly the same rules. Light particles, photons, get absorbed by molecules, named chlorophylls, which are responsible for the green color of leaves. As you learnt with the colored flames, the electrons can use energy to climb up an imaginary ladder and reach their excited state. Now here comes the difference. Instead of relaxing by jumping down the ladder and giving off light to get rid of this extra energy, they hop through a chain of other molecules. With each hop, some of the energy is released, and the leaf can use this energy to do photosynthesis and make glucose and oxygen at the end. Now that we know what happens in the leaf and how to measure how fast this happens, we can start to make a hypothesis on all the variables that can affect this process and make it faster or slower. What happens when there is less light or no light? What if there is a lot of light? What if we switch from white light to red light, green light, or blue light? Photosynthesis needs light to be absorbed so that the light energy can be used for chemical reactions. You have learnt that different colors correspond to different amounts of energy. Can you imagine what color of light would be the most effective? One of them wouldn't work at all. Can you guess which one? What if we put no sodium bicarbonate in the solution? Or what if we put more? Maybe a lot more. What happens to the floating leaves when we turn off the light after the experiment? Do you think that they will keep floating? Or will they sink? What would be using up the oxygen that they made?